So let's have a look at the development of a method section for a phase one paper. We'll use this paper on the safety, tolerability, and pharmacokinetics of senapid, hope I'm pronouncing that right, um, for solid tumors. And this is a Chinese study. Okay, so with respect to the length of a method section, it really is dependent on as long as it needs to be. Um, you can see most sections, most methods will be broke up into a study design, a patients, an objectives, and a statistical analysis section. Depending on the different target journals, these sections can be structured differently. So, um, don't forget you can include any information that can't be fitted into the text that may be stipulated due to the journal guidelines into supplementary files, as the authors have done here. So let's just have a look at the components of this particular method section. Okay, so let me just get this up. So first, so more often than not, the opening paragraph in a method section of a paper reporting a clinical trial would be a description of the trial design. And it's good publication practice to also refer to the countries in which the trial was conducted, and we can see that the authors have done this here. Okay, so this is a phase one study, and so there's distinct characteristics of this trial type that must be reported in the method section. So phase one trials, they're early stage studies primarily designed to evaluate the safety, tolerability, pharmacokinetics of a new drug or intervention in a small group of healthy volunteers, or they can be actually patients with the condition that you're assessing. Usually about 20 to 100 participants. These trials are the goal of them often is to identify the appropriate dosage levels and potential side effects, um, and they serve as the foundation for subsequent efficacy testing in a phase two trial. So, determining the safety profile of a new drug and thus the primary endpoint of phase one trials. Efficacy is more and more often brought forward as a co-primary endpoint. So generally, there are two phases or components to a phase one trial: a dose escalation and a dose expansion stage. So how the trial was designed to assess this needs to be referred to. The authors do this, it's in this blue text, in these subsequent paragraphs here. Um, so first, per this paper, we must describe the inclusion and exclusion criteria with respect to enrolling the people who are participating in the trial. Um, it's more kind of correct to refer to such characteristics as the eligibility um, criteria. Exclusion has a kind of negative connotation. So, if possible, I would recommend referring to eligibility as opposed to inclusion exclusion. So, um, the time point prior to starting the trial, when suitable patients are being assessed by the trial investigators, is referred to as the baseline, or sometimes week zero. So this is the time point at which these assessments, the eligibility criteria, will be assessed. So here the authors have noted the primary eligibility criteria for the participants in each phase of the trial. Phase two or phase three trials typically have an induction and maintenance phase, and there may be criteria for patients to progress from one to the other, um, such as response to treatment at the end of the induction phase. Um, so, with respect to the patient eligibility here, the authors have quite a short summary. Um, and that's okay because we have a supplemental file referred to here where this information is reported in more detail. So eligibility criteria they set themselves can be quite extensive. um, and this can be have an effect on this can influence the word count of your paper. So if you can include some of this information as a supplementary file, definitely do so. Um, this trial we can see here, which with respect to some specific criteria, so it's looking at solid tumors, um, and this is done so via the response evaluation criteria in solid tumors. This is the RECIST criteria version 1.1. So let's have a quick look at what these are. Okay, so the RECIST guidelines were first developed in the year 2000 by an international collaboration that included the European Organization for Research and Treatment of Cancer, the EORTC, the National Cancer Institute of the United States, this is the NCI, you'll probably come across this a little bit when you're writing papers, and also included the National Cancer Institute of Canada Clinical Trials Group, the NCICCTG. So there was an updated version, this version, version 1.1. This was published in 2009 to address some limitations in the original criteria. You'll come across these guidelines all the time when writing papers on oncology, especially those reporting on solid tumors. So it's important to have a basic familiarity with what they report. So the RECIST guidelines, they state the criteria to determine whether a tumor disappears. This is called a complete response. It shrinks. This is a partial response. It stays the same. This is stable disease. And these are gets bigger. And these are all based on measurable changes in tumor size. Computerized tomography, CT scan, is the preferred method to measure tumors at baseline and the change in size during treatment. So the baseline scan should be done within 4 weeks before treatment starts. And what physicians do is that they choose 5, maximum 5 target lesions. They're easy to measure, and they calculate this at the sum of the longest diameters. So this is an important concept just to be familiar with it. If you want some good examples of how SLD is calculated, have a look in this paper in the American Journal of Routine Genealogy. Okay, so once we've described the eligibility criteria, we next need to get into the actual study design itself. So in this paper, the overall study design is appropriately referred to in the opening sentence of the method section. Then the patient eligibility criteria are described. So now we're back to the study design section here. This could be due to the journal guidelines regarding the structure of the paper, or it could be author preference with respect to the flow of the method section. Another possibility is to separate the study design and patient eligibility criteria into distinct subsections. But as the title of this subsection is study design in patients, I think it's fine with respect to the story, it makes sense. So let's have a look at the study design. So as this is a phase 1 study, we must describe the methods used to identify the appropriate dose. So here, dose escalation was conducted using a modified Fibonacci 3+3 methods, starting at 2 milligrams once daily. So this is QD. QD, this is an abbreviation from the Latin quadra D, and escalation up to 120 milligrams across 9 dose groups. The vast majority of phase 1 studies use this 3+3 method, starting at 2 milligram once daily. Okay, that's what they did here. So sorry, the 3+3 methods, this design proceeds in cohorts of 3 patients treated at increasing dose levels. So dose escalation stops as soon as at least 2 out of 3 or 6 patients experience dose-limiting toxicities. These are DLTs at that dose level. So during phase one trials, researchers are typically aiming to find the maximum tolerated dose, the MTD, um, or the recommended phase two dose. But from a statistical standpoint, there is some controversy regarding use of the 3+3 method. See this paper for further information. So let's get back to the study design. We can see at each dose level, patient receive a single dose of sinaparib, following by a 7-day washout period before entering a continuous dosing period of daily administration from days 1 to 2 in a 3-week cycle. This was to confirm the safety and pharmacokinetic profiles. Progression to higher doses required data safety monitoring committee approval based on the safety of the PK data from previous group. So assessing drug pharmacokinetics is a key aspect of phase one trials. These are assessment of the movement of a drug into, through, and out of the body, the time course of its absorption, bioavailability, distribution, metabolism, and excretion. If people are interested in learning more about pharmacokinetics, pharmacodynamics, please leave a note in the comments. So back to the methods. At 50 milligram twice daily, so twice daily is abbreviated as BID, derived from the Latin bis and D. Dose group was added following the Data Safety Monitoring Committee review of safety, PK, and efficacy data from the once daily groups in a previous phase one trial in Australia. This is nice. It shows that the authors and trial designers were adaptive and respected their study protocol as new research became available during the conduct of the trial. It's not uncommon for trial protocols to be amended as it's conducted, the reasons for which are noted in an updated protocol and should be available on wherever the electronic record is stored on websites such as clinicaltrial.gov. So since this is a phase one study, its dose expansion period next needs to be described. So the dose expansion phase of a phase one clinical trial is designed to confirm the safety, pharmacokinetics, and preliminary efficacy of a drug at the maximum tolerated dose or the recommended phase two dose. So the goal is to further evaluate clinical responses such as complete or partial responses and assess the treatment's tolerability in larger cohorts. Patients are usually treated with the established dose regimen, i.e. once or twice daily, until disease progression or unaccepted toxicity is observed. This phase usually includes patients from the dose escalation phase, as well as potentially additional participants to expand the data set. Specific populations of interest are often also targeted, such as those with a specific genetic mutation or specific tumor types. And we see in this study, the authors ticked all these boxes. So we can see that the dose expansion period included 3 populations. Dose groups where at least one participant achieved a clinical response or a partial response per the RECIST guidelines. One dose higher group, these were evaluated for safety, and a group at the maximum tolerated or recommended phase 2 dose. So we have these 3 groups. So each group included patients from the dose escalation phase, as one would expect, and an additional planned 10 patients, resulting in approximately 20 patients in the maximum tolerated dose or recommended phase 2 dose group. Enrollment focused on patients with this BCRA positive mutation, solid tumors, and patients receive sinaparib monotherapy orally, once daily or twice daily in 3-week cycles until disease progression or unacceptable toxicity. So ultimately, results from this phase of the trial will help to refine the dosing strategy and guide the design of subsequent trials. So this is a common structure when writing up a phase 1 study, a paragraph or so on the dose escalation component, and a paragraph or so on the dose expansion period. And the authors have quite a nice succinct summary here. So this content in purple, this is respect to the ethics and oversight. So for good publication practice, ethics and oversight considerations must be reported in a paper reporting on a clinical trial. So this is often done at the end of the study design section, or it can also be included in its own subsection at the end of the trial. Where the trial is registered can also be noted here. This is usually on clinicaltrials.gov or its European counterpart. The EMA UTRA CT database is sometimes also reported. Where the ethics, where this information is reported, it may depend on the journal guidelines. They may stipulate where it has to be reported. So for the present study, we can see that the trial adhered to ethical standards, including the Declaration of Helsinki. We have some regional, i.e. Chinese-specific clinical practice guidelines and the technical guidelines for clinical trials of anti-neoplastic drugs. So if anyone is interested in learning more about these different types of guidelines, do leave a note in the comments. We see that all participating patients provided informed consent. The NCT number is reported. Okay, this is nicely summarized in a succinct paragraph. So a key component, as one might expect, in reporting certain information in the method section is what were the study primary objectives and endpoints. So where these are structured is going to depend on the journal guidelines. So after the eligibility and study criteria information is reported, we next go on to this section. Researchers conducting phase 1 trials like this are often interested in a couple of different outcomes. So as these phase 1 trials are often the first time a drug was assessed in humans, the researchers conduct assessments to collect as much information as possible, and this information will guide future trials. So here the authors open with the study objective. The study objective is sometimes reported in the first paragraph of the methods. It's also often reported at the last few sentences of the introduction section, except that it's usually expanded upon a little bit in the methods. So we can see the trial aimed to evaluate safety, tolerability, pharmacokinetics, and preliminary anti-tumor efficacy in the drug, as well as establish the maximum tolerated dose. This is a standard objective for a phase 1 study. So let's have a quick look at the primary paper and let's see if they did indeed report this in the introduction. I believe they did. Okay, we can see here at the end of the introduction, a phase 1 first-in-human study was conducted to evaluate the safety, tolerability, and PK profile and anti-tumor activity of sanaparib in Chinese patients with advanced solid tumors. So this objective was further expanded upon in this first paragraph here. Okay, great. So let's get back to objectives here are for our objectives and outcomes assessment. So then we get to the outcomes after we stated the objective. You can see here they start off with safety. Safety was evaluated by tracking dose-limiting toxicities and treatment emergent adverse events per certain criteria. These are the NCI-CAE criteria. These are pretty standardized for reporting adverse events in clinical trials. So safety assessments are often reported themselves in their own subsection within the methods, and this is often the case for papers reporting the results from phase 2 or 3 trials. So consider this approach depending on the amount of safety information you wish to report in the methods. We also, so moving on, the maximum tolerated dose determination. That is defined here. So this is defined as a maximum dose at which no more than 1 out of 6 patients experienced a DLT. The pharmacokinetic assessments or the parameters were analyzed. We can see from blood samples collected after a single dose and throughout the first treatment cycle, with plasma concentrations measured using high performance liquid chromatography, tandem mass spectrometry. Okay, so we can see that once we describe the PK, the authors have a brief description on the tumor efficacy assessments. So we can see tumor response was evaluated every 6 weeks using the RECIST guidelines, focusing on objective response rate, disease control rate, duration of response, and PFS, progression-free survival. And they included patients with this particular mutation. Okay, so our final section, all method sections in all clinical trial papers must include a description of what statistical analysis were performed to collect the data. So these will vary depending on the trial designs and the outcomes being assessed, but they will be planned in advance and described in detail in a statistical analysis plan, a SAP. The SAP will typically be the source the medical writer will use to develop the statistical analysis section. Typical things that are reported in this section include how the study sample size was determined, what analysis sets were used, and how these were defined. So the safety set, the full analysis set, the intention to treat. Sometimes there's a modified intention to treat set. How these were all defined and analyzed is a key component to report. Here we can see they're going into more information in the supplementary materials. The statistics used to estimate treatment effects should also be mentioned. Here we can see that Kaplan-Meier statistics were used. These are stats commonly used to affect treatment effects over time. We should also refer to whether probability or descriptive statistics were used and what range was used to estimate the parameters being investigated. Here we can see the researchers used 95% confidence intervals. Assessment of pharmacokinetic data often include area under the time concentration curve, AUC, and maximum plasma concentration analysis, Cmax here. So the area under the time concentration curve measures the total amount of a drug in the bloodstream over time and showing how much of the blood that a body absorbs, whereas the Cmax, the maximum plasma concentration, this is the highest level of the drug in your blood after taking it and indicates how quickly it reaches peak levels. So these are key analyses in phase 1 and 2 trials. It's quite often to include additional information in the supplementary stats section, as I mentioned. It's also good practice, and it's quite often journal policy to indicate what statistical software was used to perform the analysis. We can see here that SAS, this is quite a common one, was used with a different software used for the PK analysis. If you're interested in hearing more about the development of different types of method sections for clinical trials, do please leave a note in the comments and I'll do my best to get back to it. To conclude, we've examined the 5 key elements of the method section for a clinical trial paper using this phase 1 study as an example. These key elements are describing the study design, this text in blue, the patient eligibility criteria in green here, some information on the ethics and the trial oversight here in purple, a description of the study objectives, outcomes, and endpoints, reference to the statistical analysis, And that's it. These are all the critical components that should be referred to in the method section of any paper to align with good publication practice. So it's important to note that the order that each element is reported can vary depending on journal guidelines, the type of trial being reported, or even author preference. It's good practice, though, for the method section reporting a clinical trial, as I said, to mention each of these elements. So I appreciate if you can click that like button if you found this video helpful or interesting, and if there's any particular topics that you'd like covered in the world of medical writing, do please leave a comment, and good luck.